cover chapter 10, which is basically about DNA, the genetic material. We're going to cover DNA replication, transcription, and translation. Before we go into this chapter, I want to let you guys to please note the scientists' names and the significance of their experiments. So I know it's been a long semester with these online videos, but we are almost to the end, so please just hang in there. Let's review about the DNA structure. So we know in the beginning of the semester, we talked about how there's two DNA strands in one molecule. And then these two strands, what they do is that they coil together, and then it'll form what's called that double helix. And then if you unfold or uncoil this double helix, what you're going to see is that it'll appear like a ladder. And then this ladder, the sides of the ladder, you'll have your sugar phosphate backbone. So sugar phosphate, etc. So these are the sides of the ladder. And then what's attached to the sugar phosphate backbone is a nitrogenous base of one strand that will bond to the nitrogenous base of another strand. And then so here you have adenine bonding with thymine, and these are bonded through hydrogen bonds. And then recall, hydrogen bonds are weak bonds, so that means that they can break and reform easily. So that's really important when we want to carry out replication, DNA replication. We want to break this double helix, break these two strands, and then when we finish replication, we can just rebond the new daughter strand and the parent strand easily. And then we also know that we have adenine will always base pair with thymine, whereas cytosine will always base pair with guanine. And then these are how these two strands are connected together. All right, let's get to our first experiments. So in the 1950s, before then, nobody really knew what was being passed down. What was the genetic material? What was the hereditary material? Was it the DNA or the protein that was being passed down, injected, or being copied? Well, Hershey and Chase, what they did is that they had an experiment with viruses that specifically infected bacteria, and then these are called bacteriophages, or phage for short. They used these two organisms to illuminate or discover that it was the DNA that was being passed down. Let's take a look at the structure of a virus. So all viruses will contain two components. They have the nucleic acid, and some viruses will have DNA as their nucleic acid, and then others will have RNA as their nucleic acid. Well, how do they protect this nucleic acid? They have a protein coat surrounding it. So there's one virus with protein coat, and then here's another with a protein coat. And then, a third component, which some viruses will have, are these spikes that have glycoproteins, and then these will attach to other cells or surfaces. So basically, how their experiment is run, you have your bacteriophage, a virus that was specifically infect a certain bacteria. So here's, the virus attaching to the bacteria, and then we know that the virus will inject its DNA into the bacteria, and then the DNA will use the host's, the bacteria's machinery, cell resources, and energy to make new parts of the virus. Okay, and then what will happen is that these parts will assemble into new viruses. So we'll have viral progeny, and then when these viral progeny are ready to infect other bacteria or other hosts, they are going to break free, ultimately killing their host or that bacteria. So to get into their experiments in more detail, they ran two batches or two experiments. So the first one, what they did is that they had this bacterial phage, specifically called a T2 because that virus specifically infected E. coli. For the first batch, what they did is that they radioactively labeled the protein in the protein head of the virus. Okay, they radioactively labeled the sulfur to glow a bright yellow color. And then so they allowed the virus to infect the bacteria. So when it does, okay, it does its thing, it infects, and then they put it in a blender to agitate so that the protein coats would detach from the host, the bacteria. And then they place it in a centrifuge. And then what happens when you place things in a centrifuge is that the material that is heavier will go to the bottom, will settle to the bottom, where the lighter material will appear at the top of the centrifuge. So in this case, what happens is that the protein shells, or the protein coat of the virus that was radioactively labeled yellow, stayed at the top, whereas the bacterial cells, which are heavier, went to the bottom. And then we can see that the bacterial cells weren't actively labeled. It was the protein shells. So that means that the protein did not enter the bacterial cells. Now, they proved that it was the protein that didn't enter into the bacterial cells. So that means for their second experiment, they had to prove that it was the DNA that entered into the bacterial cells. So they ran another batch for these T2 bacterial phages to infect the E. coli. So what they did is that they radioactively labeled the DNA, which has a phosphorus, green. And then you can see that it's radioactive labeled green, okay? And then they let that infection occur. And then they also put that into the blender to agitate, to detach, to remove those protein shells. And then again, they placed everything in a centrifuge where the heavier component, the bacterial cells, will go to the bottom. And then you can see that now the bottom, the pellet, it actually is the one that is radioactively labeled. It appeared green, so that means that the DNA entered the bacterial cells to make it a green color, whereas your protein shells, the protein coats, are not radioactively labeled. They didn't enter the bacteria. So this is how they proved that it was DNA that is the genetic material that was used to replicate things and to that entered the bacteria. Another experiment that was carried out by Watson, Crick, and Franklin, although Franklin didn't get the notoriety that Watson and Crick did. So what happened is that these three scientists, they proved how the DNA structure was, how the shape was, okay? What happened is that Franklin, she used the X-ray crystallography experiment to prove how the atoms were arranged in the DNA molecule. What they did is that they had an X-ray source and then they crystallized a DNA sample, and then they used a beam of X-rays that will hit that crystallized DNA structure sample. And then when it does is that it makes a map of the atoms, specifically the electrons in the DNA molecule, to form a pattern. And then from this image, from this pattern, they were able to discover that DNA was in the form of a double helix. It has 2 polynucleotide strands. Your nitrogenous bases are stacked on top of each other. And then there is a uniform diameter. So the diameter is the same throughout the DNA structure. So let's talk more about the DNA structure. So we already know that it has a sugar phosphate backbone, okay? And then here we have what's called a 5' N and a 3' N. So here's one strand, here's another strand. Well, each strand of DNA runs from the 5' N to the 3' N. What does that mean? So here is the sugar of your nucleotide. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Okay, and then so at the fifth carbon at the 5' N, the fifth carbon, there's a phosphate attached to it, a phosphate group. So when you hear 5' N, I want you to associate phosphate attached to that carbon. And then at the other end of the strand, the same strand, you have a hydroxyl group attached to the third carbon. So carbon 1, carbon 2, carbon 3. So this hydroxyl group, this OH, is attached to the 3' N. So that's why it's called 3'. So 5' N phosphate, 3' N hydroxyl group. Tied with this phosphate at its 5' N will be attached to the hydroxyl group at the 3' N. So carbon 1, carbon 2, carbon 3, which will have a hydroxyl group attached to that phosphate. So that's how these nucleotides are attached to each other. So that's one strand going from 5' to 3' N. Well, the other strand, it has to go from 5' N to 3' N, but it'll go in different directions, opposite directions. So we call that anti-parallel. So here, this end of this strand, we have the fifth carbon right here with its phosphate group attached to it. And then here at the other end of that same strand, we have the hydroxyl group attached to the 3', the third carbon. And then polarity, it just means that is different on the ends. And then this is just another review of the DNA structure. We know that DNA is made up of monomers or subunits called nucleotides. And then these nucleotides, they have a 5 carbon sugar. So 1, 2, 3, 4, 5 carbon sugar. And sometimes you'll hear it as a pentose sugar, pentose sugar. And then also you have your phosphate group and then your different nitrogenous bases. Well, these nitrogenous bases, we can categorize them based on their structure. So if the nitrogenous base has a single ring, they are called pyrimidines. And then these will include your cytosine and your thymine in DNA. And then when you think of thymine, think of uracil in RNA. So these three are classified under pyrimidines because they are single ring structures. And then one way to remember is that pyrimidine has a Y in it, cytosine has a Y in it, and then thymine has a Y in it. And when you think of thymine, think of uracil. The other type of nitrogenous base you can have are called purines, and then these are bigger, these are double ring structures. So one ring, another ring. And then these will include your adenine and your guanine. And then one way to remember is pure as gold. Purine, adenine, and guanine. And then we know that a purine will always bond with a pyrimidine. So two purines will never bond with each other. Two pyrimidines will never bond with each other. Another scientist is called Chargaff, and then he made up the rules of the base pairing, which you guys already know. We know that adenine will always base pair with thymine, And then guanine will always base pair with cytosine. So that means that the amount or the number of adenine will always equal to the number of thymine. And then also the number of guanines will always equal to the number of cytosines. So ultimately, we can always refer or, include that purines, the number of purines will always equal to the number of pyrimidines. And then there's this equation that you can use to find the amount of a certain nitrogenous base. And then here is an example. So let's take a minute to find out what the percentage of cytosine is if we know the number or the percentage of adenine. I'll give you guys a minute. Okay, so based on chargaff, we know that, if the amount of adenine is 15%, then thymine has to have an equal percent as well because they always base pair with each other. So adenine is 15%, thymine is 15%, okay? So you add those two together and you get 30%. Well, the rest of the DNA sequence will be made up of guanine and cytosine. So what you do is you minus that 30% from 100, and then you get 70%. And then we know that the amount of cytosine always equals the amount of guanine. So what you do next is just divide it by 2. And then we get 35% is guanine, and then 35% is cytosine. Now let's compare the structure of DNA to RNA. So one of the main differences between these two molecules is that DNA, okay, its sugar is called deoxyribose, just because it's missing an oxygen from the second carbon. So carbon 1, carbon 2. So here, this is missing an oxygen, so that's why it's called deoxyribose. Deoxy means without oxygen. Whereas the RNA, its ribose sugar, does have an oxygen at the second carbon. So carbon 1, carbon 2, it does have an oxygen here, ribose sugar, deoxyribose sugar, missing an oxygen. And then we also know that the DNA molecule is double-stranded. It has 2 strands, whereas the RNA is single-stranded. And the RNA does have a sugar phosphate backbone along with those nitrogenous bases. But the different nitrogenous base that RNA uses is uracil, and then DNA uses thymine. So those are some differences among DNA and RNA. But for this test, you guys will have to know the similarities between them and then the differences between those two. So this is just a chart that I've made for you guys. So some similarities is that both molecules are made up of nucleotides. They have nucleotides as their monomers. These nucleotides can come together to make polynucleotides. Each of these nucleotides for both molecules will have a 5-carbon sugar, or called a pentose, phosphate group, and a nitrogenous base. They both have sugar phosphate backbone. They both use guanine, cytosine, and adenine, and then they're both found in the nucleus at one point. Some differences here is that DNA contains a deoxyribose sugar, whereas RNA contains ribose sugar. And then DNA uses thymine that will base pair with adenine. RNA will use uracil that will base pair with adenine. DNA is double-stranded, RNA is single-stranded. DNA is so important that it never leaves the nucleus. It always stays in that nucleus. Well, RNA, as soon as it's transcribed, it goes out of the nucleus into the cytoplasm to meet a ribosome to get translated. We only have one type of DNA, whereas RNA, we actually have 3 types, which we're going to talk about all of them in this chapter. We have mRNA, rRNA, which is the ribosome, and then tRNA. DNA is involved in replication. So we have DNA replication, and then DNA can be transcribed into mRNA. And then we have our mRNA that was made from transcription. And then it gets translated into amino acids to make our protein. And then our DNA is way much longer than our RNA. Our mRNA is the length of 1 gene, basically. There's this chart on Blackboard. It's a good idea for you guys to complete it to keep things organized. You know, what's involved in DNA replication, what's involved in transcription and translation. Because I think that would be one thing that, you know, students mess up on the exam is keeping it organized. Okay, so now let's talk about DNA replication. So what phase does DNA replicate in? You guys remember from chapter 8 is that S phase, that synthesis phase, in interphase. So that's when DNA replication occurs. And then there was 3 models that was proposed for how DNA replicates, and it's this model, semi-conservative replication, that Messelson and Stahl, the scientists, prove that that's the way DNA replicates. So what happens is in semi-conservative replication is that you have the parental strands. So you have these 2 parental strands that make up the DNA molecule, and then what's going to happen is that they will disconnect, and then this one parental strand will serve as a template to make this new daughter strand, and then this will be one new DNA molecule, whereas the other parental strand here will serve as a template strand to make the new daughter strand for another DNA molecule. So each new DNA molecule is half parent and half daughter strand. In DNA replication, you have to have like a start point where DNA replicates. That is called the origin of replication. So in prokaryotes, bacteria, they have a singular circular DNA chromosome. And then since they don't have that many genes, they only have one origin of replication where these 2 strands will disconnect. So that's your origin of replication. And then what happens is the bubble will get bigger and bigger, and then you'll have replication occurring in this direction, and then replication occurring in this direction to be more efficient. So when it proceeds in both directions, we call it bidirectional. So prokaryotes, the DNA replication occurs in the cytoplasm, or that nucleoid region, because they don't have a nucleus. Whereas us, eukaryotes, our DNA replication occurs in the nucleus because that DNA never leaves the nucleus. And then since we humans, we have basically 3 billion base pairs, if we only had one origin of replication, it would take so long to replicate our entire genome. And then cellular division would take very long. So for us, we have many origins of replications. So here's one origin replication, here's another, here's another. And then when these strands open up, we form what's called a replication bubble. So here's a bubble, here's a bubble, here's a bubble. And then just like in the prokaryotes, our replication is bidirectional. We'll have replication occurring this way and then occurring in that way. So that way our entire genome gets replicated in a much faster and more efficient way. So for DNA replication, we actually took a lot of material out for you guys. So in your book, they talk about DNA primase, DNA ligase, Okazaki fragments, leading and lagging strands. Don't worry about any of those. So just worry about what's in your lecture notes and then what we talk about here. Okay. So here you have your DNA double-stranded double helix. And then the first thing that's going to happen is that you're going to have an enzyme called DNA helicase. DNA helicase. And then what it does is that it will break those hydrogen bonds to separate the two parent strands. So we basically say that this DNA helicase will separate, unwind, disconnect these two parental strands. And then next we have another enzyme called DNA polymerase. So this DNA polymerase, it is the enzyme that will add the nucleotides, that it will make the new daughter strand of DNA. And then this DNA polymerase, what it does is that it will read the parent strand from the 3' direction to the 5' direction, so that DNA polymerase can make the new daughter strand from the 5' direction to the 3' direction. So basically this DNA polymerase is kind of like reading the template strand backwards. So remember how I said, our DNA strands go from 5' to 3' direction? Well, this DNA polymerase is reading it backwards, like it has dyslexia. So it's reading the template 3' to 5' so that it can make the new daughter strand 5' to 3' direction, the correct direction. And then this DNA polymerase, another thing it does is that it can only add new nucleotides to the 3' end only. So remember, the 3' we have a hydroxyl group attached to it. So here's the existing strand, and then here we have the new nucleotide. Well, it can only add to the 3' end only. And then here in this picture, you can see that our template, well, it runs 5' to 3', but our DNA polymerase will read the template 3' to 5' so that it can make the new daughter strand 5' to 3' direction. So our DNA polymerase will go in this direction here. And then after it adds the nucleotides, it has the ability to proofread. So if there's a nucleotide that was mistakenly put that doesn't match the template nucleotide, it'll take that out and then put in the new one. So for example, we know that cytosine is supposed to base pair with guanine. Well, what happens if it accidentally put an adenine? Well, the DNA polymerase is going to proofread that, recognize that that's the wrong nucleotide. It shouldn't be an adenine there. And then it'll take that out and then put a cytosine that will correctly base pair with guanine. And then also what you have is when your DNA helicase unwinds the helix, you have this strand opening up from this strand that's separating, and you get what's called a replication fork. Replication fork kind of resembles a utensil fork. How do we get from the DNA molecule to our amino acid sequence to make our protein? One of the general principles we've been talking about throughout the semester is that how do we, we start off with our DNA, our gene, which is double-stranded. Okay, so this is our genetic makeup, our genotype. And then when we need a protein made, like pigments, that DNA gets transcribed into a molecule called mRNA. The M stands for messenger RNA, and then this is single-stranded, and this happens in the nucleus. Next, that mRNA, it leaves that nucleus through the nuclear pore, and it goes into the cytoplasm to meet with a rRNA. This R stands for ribosomal RNA, which is basically your ribosome. It meets with the ribosome to be translated into our amino acid sequence. And then this happens in the cytoplasm where it meets the ribosome. And then another player that we're going to talk about is tRNA. The T stands for transfer RNA. And then with all these three RNA molecules, we're going to be able to make our protein, our pigment, when it needs to be expressed. So remember, what we see is our phenotype, that trait. So this is the process, this is the principle, how we get from our genotype, our DNA, our genetic makeup, to what we physically see, what is observed, the trait, which is called our phenotype, which is the protein. And then so that's where everything happens for us, for the eukaryotes. But for the prokaryotes, since they don't have a nucleus, everything basically happens in that cytoplasm, where you have its DNA is transcribed into mRNA, and then it'll meet with the ribosome as well to be translated into our amino acid sequence, or a long string of amino acid sequence called polypeptide. And all of this occurs in the cytoplasm for bacteria. Okay, so here the next process that we're going to talk about is called transcription. So this transcription is how we go from the DNA, it is converted into a similar language called that mRNA, messenger RNA. And then we just stated that it happens in the nucleus. So one of the key players in this process is RNA polymerase. RNA polymerase, not DNA polymerase. So this RNA polymerase, what it does is that this enzyme will find a sequence on the DNA that is called the promoter. And then this is a signal to where the gene starts. And then this RNA polymerase, what it does is similar to DNA polymerase. It reads the template from 3' to 5'. So in the backwards direction. So it reads this template of DNA from the 3' to 5' direction so that it can make the mRNA in the correct direction of 5' to 3'. So this RNA polymerase, this enzyme, it'll add the nucleotides but it's RNA nucleotides. So RNA nucleotides, we still have adenine, guanine, you don't see it, but cytosine. But in RNA, we have uracil that will base pair with the adenine instead of thymine. So remember, in RNA, we have the uracil base. So this new mRNA, it runs in the 5' to 3' direction. So it keeps adding and adding and adding. And then as that mRNA is made, it peels from the DNA molecule. And then RNA polymerase knows to stop where the gene stops at this terminator DNA sequence. So this is the stop signal, this terminator sequence for the RNA polymerase. And then once it reaches the terminator DNA sequence, that RNA polymerase will be removed or detached from our DNA molecule. And now you have your mRNA that will carry the instructions to make the protein from the nucleus into the cytoplasm. Okay, so the next process is translation. And then this translation is when you are changing that mRNA language into a different language of amino acid sequence. And then a long string of amino acid is called a polypeptide. And then these polypeptides will fold together to make your protein. And then something important to know that everything is basically read in threes. So you have your DNA read in threes, you have your mRNA that will be read in three bases at a time. That's called the triplet code. Triplet code. And then so here, in this picture, we have your DNA template. This is used to make the mRNA. And then what you have, what's going to start translation is something that's called a start codon. And then each codon on this mRNA is comprised of 3 bases. So this is a codon, this is a codon, this is a codon, this is a codon, etc., etc. So you have the start codon that will tell the ribosome where to start translation. And then you have a stop codon to tell the ribosome when to stop translation. And then this chart here is for us how to read what each codon specifies for. So each codon, each 3 different bases, it will specify for a specific amino acid. So this AA stands for amino acid. So take, for example, how do we read this chart? So here is the first base, second base, third base. So how do we know what amino acid this AUG specifies for? Well, A is the first base. So we're going to find along here where's A. So A is here. So it could be anywhere, in this row right here. And then U is the second base. So we go up here, we find where U is. So U is right here. And then that means that our codon is somewhere in here. And then G is the third base. So in this row, we're going to find where G is. And then G is on this row all the way over here in this box. So here's our AUG. This is where our start codon is, and it'll specify for methionine. Methionine. So in real life, every amino acid would basically start with the methionine amino acid. But for your exam, your homework, practice worksheets, it doesn't necessarily have to start with methionine. It can start with any other amino acid. So if it doesn't start with methionine, don't worry. It doesn't mean that it's not correct, your answer. So that's the correct way to read this codon chart, this mRNA chart. But basically, you can use that proper way, or you can just kind of scan around and see where you can find AUG. On the exam, this chart will be provided for you, so you don't have to memorize anything on this chart. Basically, you just have to know how to use it as efficiently as possible, of course. So here we see that, for our amino acid sequence, we started off with methionine. Let's look for what the next codon specify, which amino acid it will code for. So we have AGC. So why don't I give you guys a minute to find where AGC is. So A is the first base, so it's along here. And then G, the second base, will be here. So it's this column, this box right here. And then C is the third codon, or the third base. So here is C. So here's AGC, and then it will code for serine. So here we have serine. So that would be the next amino acid, and then so on and so on. And then what you have to help carry those amino acids, which we're going to talk about in a little bit as well, is something that's called the tRNA. And then on the transfer RNA, you have, on the opposite side, you have what's called anticodons. And then these anticodons are just complementary to the codon. So A, will base pair with U will base pair with A, G will base pair with C. So this is how, tRNA will recognize the codon so that it can bring the correct amino acid, so that we can have our correct amino acid sequence to make our correct protein. So on this chart, you have one start codon that will specify for methionine, and then you have 3 stop codons. These stop codons do not specify for any amino acids. But this chart is very special. You can only use it when you have the codons, when you have the mRNA sequence. That way you can find the amino acid sequence, the polypeptide, the peptide, eventually the protein. If you only have DNA, you can't use this chart yet. If you have DNA, you will have to transcribe it into mRNA, and then you can use this chart. If you have the sequence of tRNA, the anticodons, you still can't use this chart yet. From the tRNA, you'll have to find the mRNA, and that's just the base pairing rule. And then after you have the mRNA, then you can use this chart. Why do I mention this? Because students in the past, that's what they've done. They, look at the DNA and then they'll try to find the amino acid sequence already. Or they'll have the anticodon sequence, the tRNA sequence, and then they'll look on this chart and then they'll find the incorrect amino acid sequence. And then another special thing about this chart, this mRNA chart, is that each codon will only specify for one amino acid. So this UUA will only code for leucine. UUA will not code for anything else. But this leucine will have 2 codons that will specify for leucine. That's very special, because what if our original codon was supposed to be UUA, but that third base mutated or changed into a guanine. And now we have UUG, but that's okay because UUG will also code for leucine and will still have the correct amino acid sequence. And then you can see here, well, there's these 4 codons that will also specify for leucine. And then in total, there's about 20 different amino acids. So you have phenylalanine, leucine, serine, tyrosine, cysteine, tryptophan, arginine, etc., etc. So don't worry about this chart. It will be provided for you during the exam. Okay, so there's a worksheet that I made for you guys, a part 1 worksheet, and then it's really important that you know how to do it, how to complete it, because it will be on your exam. So here, let's start off if we have a situation, is given the DNA sequence. So here we have our DNA sequence made up of these nucleotides, and then it can ask you to find the amino acid sequence, the polypeptide, the peptide, the protein. So when it's asking for any of those, basically what we want is the amino acid sequence for you to use this chart and find those amino acids. So we have this DNA sequence, and then our next step before we can use this chart is to find the mRNA sequence. Well, since we have a 3' here, our mRNA, it has to start with our 5' end. And then now we just use the base pairing rule to find the sequence. So what will base pair with thymine? Adenine. What will base pair with adenine? Uracil. So remember, we are making our RNA, our mRNA, so we have to use uracil now. And then next we have G, etc., etc. And then here, since the DNA sequence ends in the 5' end, well, our mRNA has to end in the 3' end. And now we have to use this chart. We are able to use this chart now because we have our mRNA codons. So this is one codon, this is another codon, and then this is another codon, groups of 3, the triplet code. So here our AUG will specify for methionine, AUG. And then CGG, if you use the chart, it'll code for arginine. And then this codon GUA, using the chart, it'll code for valine. So our amino acid sequence goes methionine, arginine, and valine. It goes in this direction. So that's one example. Another example is, what if we gave you the sequence of the anticodons, that tRNA sequence? So here, this is our tRNA sequence. It goes from the 3' to 5' direction. Well, we have to find our mRNA, our codon sequence, before we can use this chart. So here, since the tRNA starts in the 3' direction, we have to make our mRNA in the 5' direction. It has to be anti-parallel. And then from there, we use the same base pairing rule. So what will base pair with uracil? Adenine, and then here guanine, etc., etc. And then since our tRNA ends in the 5' direction, our mRNA has to end in the 3' direction, anti-parallel. So now we have our mRNA sequence, and we can use this chart to find our amino acid sequence. And then what we find is, this codon specifies for lysine. This codon here specifies for histidine. And then this codon here will specify for cysteine. And then our amino acid sequence will go in this direction, reads lysine, histidine, and then cysteine. So please, I really encourage you guys to try to complete that chart. Now, the last process that we're going to basically talk about in chapter 10 is translation. So translation, we know, is how that mRNA carries directions to how to make the protein. Well, that mRNA will be translated, converted into a different language made up of amino acids so that we can make our protein. There's some key players. One key player is that tRNA, that transfer RNA. And then this transfer RNA is basically an interpreter of that mRNA instructions on how to make the protein. So a couple of things about the structure for the tRNA. Basically, this tRNA is single-stranded, because we know RNA is single-stranded. But what happens is that in some parts of that tRNA strand, you'll have some hydrogen bonding that occurs. So hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond. So it kind of folds up on itself. And then one end, you'll have where the amino acid will be carried by. So here the amino acid will attach here. And then at the other end of your tRNA, you'll have the anticodon. So remember, the anticodon is complementary to that codon. So you'll have 3 bases here. So this tRNA, it has 2 responsibilities. It recognizes the codons on that mRNA. So here, this tRNA with its anticodon, it recognizes its complement, this codon here. And then this tRNA, because of that, is able to carry and pick out the correct amino acid to bring for translation. So in this example, this is a very simplified picture. So here we have our ribosome, we have our mRNA, and then here you have an existing tRNA that was there already with its growing polypeptide. So here you have amino acids. And then based on this codon, the tRNA will recognize it because of its anticodon, and it'll allow the tRNA to bring in the correct amino acid to attach to the growing polypeptide to make it even longer. Your next important player for translation process are ribosomes. So your ribosomes, or your rRNA, ribosomal RNA, sorry. It has a very, very important job. It is the one that actually reads the mRNA. So it will be the one to read all the codons. So let's start from the beginning. So for your ribosome, you have a large subunit right here, and then you have a small subunit. The first thing that's going to happen is that the small subunit is going to attach to the mRNA strand. It reads the mRNA strand. It reads where that first AUG, that first start codon is. That small ribosomal subunit is going to attach to the start codon. And then you're going to have that ribosome is going to tell the first tRNA to come with its first amino acid, methionine. And then it'll be in position. So imagine if this was AUG, and then this is the first tRNA with its anticodon, and then its first amino acid, methionine. And then what's going to happen next is that you have the large ribosomal subunit attach. So that comprises of the initiation phase. And then next, once you have that first amino acid, what's going to happen is that this whole ribosome is going to move forward and read the next codon in line. And then it's going to tell this tRNA with its anticodon and with its correct amino acid, you're next. And then this ribosome is going to connect or bond the amino acids together through a peptide bond. So this amino acid is bonded to this amino acid by peptide bond. This amino acid is connected to this amino acid by peptide bond, etc., etc. And then remember, these peptide bonds are strong covalent bonds, and we want that because you want your protein intact. You don't want these peptide bonds to be weak and be able to break apart easily. So since it's the ribosomes that make these peptide bonds, that's why we say it's the ribosomes, the rRNA, that actually makes the polypeptides, that actually makes the proteins. So this ribosome, it'll position the mRNA in the correct place, the correct codon, and then it'll bring in the correct tRNA so that the tRNA can bring in the correct amino acid. So our ribosome will know where the gene starts, where to start translation by recognizing the start codon, by reading the start codon. And then it'll read along the mRNA, and then it'll reach the stop codon, and that's when the ribosome knows to stop translation, So remember, we have 3 stop codons that do not code for any amino acids. Okay, so now we are finished with those 3 very important processes. What we're going to talk about next are mutations. So we basically talked about mutations before, really in the beginning of the semester. We know that mutations is when even one nucleotide or several nucleotides are changed in a DNA molecule. And then when that happens, what we get is when that DNA gets transcribed into mRNA, we get different codon. And then sometimes that different codon will lead to a different amino acid. And then different amino acid sequence can lead to a different protein made. Now, we always, think about how mutations are bad. Yes, there are mutations that will cause disease. But, mutation is the original source of evolution. Sometimes mutations can make proteins better, can make the shape better so that it'll allow the protein to function better. So mutations aren't always bad. And then these mutations, they can occur during DNA replication when that DNA polymerase fails to recognize, a wrong nucleotide that it made, inserted in. Or mutations can occur by physical or chemical agents. So physical agents, we have X-rays, or from the sun, UV rays, drugs, alcohol, chemical agents. So we just talked about how if, we had a different codon. So for example, our original codon was supposed to be UUC, and that was supposed to code for phenylalanine. But the C, the cytosine, mutated to adenine, and now, what was supposed to be phenylalanine, we have leucine. So that changed into a different codon, a different amino acid, and now definitely a different protein. and it could be good or bad. So for mutations, a good practice is to complete part 2 worksheet, and that's posted on Blackboard as well, along with the answers. So mutations, they can fall into 2 categories. You can have base substitutions, where you can have a wrong nucleotide put in, or a group of wrong nucleotides, and sometimes these are called point substitutions. Or you can have another category where a nucleotide is inserted or deleted out. Let's talk about the base substitutions first. So there's 3 types. One type we've actually talked about, something that's called silent mutations. And then these silent mutations occurs when you have a codon change, but ultimately the amino acid sequence will be the same. So example, again, here, UUA, well, what if this third base mutated to a guanine, a G? Well, that's okay, because we'll still have leucine as the amino acid. And then our protein will be the same. So we'll have same function. Or even serine, where the third base changes to any of those 4 nucleotides, bases, it's okay, because we'll still code for serine and we'll still make the same protein. Now here, missense mutation, the other type, the codon, the DNA being mutated to make a different codon, well, that will actually change the amino acid. And then a good example, which we'll talk about, is sickle cell anemia. So here, for example, again, if this was supposed to be UUC, supposed to code for phenylalanine, but this third base mutated to be adenine, well, it'll code for leucine instead. So that's a different amino acid, different protein. And then an example is your sickle cell anemia. So here, we've talked about sickle cell anemia before, where that hemoglobin, that protein, it has a mutation, and that mutation causes the hemoglobin to crystallize, and that changes the shape of your red blood cells to be crescent shape or sickle cell, and then that'll give a slew of problems, health problems. But let's look at it on the molecular level. So here we have our normal hemoglobin protein DNA sequence. Part of it, here is supposed to be CTT, and then the correct codon for that would be GAA. And then that GGA, if you look on the chart, will code for glutamate. But however, people with sickle cell anemia, they have 1 point mutation. So here, now you have the DNA as CAT. Well, this A was supposed to be a T, but it mutated to an A. So now the CAT, the codon, that RNA polymerase, will make the mRNA as GUA. Well, now this GUA codes for valine. So it was supposed to be glutamate, but the amino acid sequence changed. Now it has a valine instead. And that 1 point mutation will cause the sickle cell anemia. So that's an example, that's a really good example of a missense mutation. So the other type of mutation that you can have is called nonsense mutation. So this nonsense mutation, what happens is that it changes the amino acid into a stop codon. So remember, our stop codons do not code for any amino acids. So imagine if this occurred right in the middle of our mRNA. So that means you only have the first half of your amino acid sequence, and you're missing the entire second half of your amino acid sequence. If that happens, you the protein that is made will definitely not work. What you get is called a truncated polypeptide or truncated amino acid sequence. It stops prematurely. Take, for example, here we have UAC. Well, what if this third base somewhere in the middle of the mRNA, this third base, this codon was supposed to code for tyrosine, but this third base, it mutated to an adenine, where now you have UAA, and then now this ribosome will recognize that UAA as a stop codon, and it'll stop translating. And that means that the ribosome has not finished translating the rest of the mRNA. So a really good example of this nonsense mutation is cystic fibrosis. So in cystic fibrosis, we talked about throughout the semester, is when you have a thick mucus buildup. It's where, these patients, they don't make the correct protein, the correct chloride channel. And then where chloride's supposed to go, water is supposed to follow. So that means, their mucus will be thick. They will get a lot of bacterial infections. They won't be able to breathe. They're in and out of the hospital. Many of this we talked about. So what happens is, here in a normal patient, here we have our DNA molecule. And then this one is the template. Here, so this one's the template that we are supposed to use to make our mRNA. So this is supposed to be guanine, will base pair with cytosine. And then this codon will make glutamine. But here, when a nonsense mutation occurs in a patient that has cystic fibrosis, what happens is that this DNA strand here, this top one, well, this cytosine got mutated to a thymine, and then thymine base pairs with adenine. And then this template now is different. So now, what will base pair with adenine to make the new mRNA strand is uracil. And then UAG, well, UAG codes for a stop codon. So now that ribosome will think that the protein, the amino acid sequence needs to stop. So originally it was supposed to make glutamine, etc., but the amino acid sequence stopped prematurely. Because now that codon, it codes for a stop codon. So now the next category, we have situations where bases will be inserted in or taken out. Let's look at both situations. So we know that everything is read in threes, the triplet code. So this is a codon read in 3. And then this is a codon, and codon. And then each codon will specify for a specific amino acid. So here we have our normal mRNA to make our normal protein. Well, what happens if we insert or put in an extra base, nucleotide? So when we do that, what we're going to do is that we're going to shift all these nucleotides downstream from here, down. So everything downstream after the insertion will be regrouped. So here, let's start at the beginning. So here we have our normal start codon, methionine. Here another codon, normal, lysine. But here, this is when the change occurs, when we get a base insertion. So now what we're supposed to have was UUU that codes for phenylalanine, but now a G is inserted in. Now it's going to read UUG. This codes for leucine. And then the next set of codons will be regrouped. So the next set originally was supposed to be GGC, but now is UGG, and that will code for tryptophan. And next we have this codon that codes for arginine. So these base insertions, they also categorize as frame shift mutations because it alters that reading frame. It regroups, it shifts everything downstream. How we read these codons is different now. We were supposed to read it like this here, but now we read it like this. Now, would this qualify as a missense mutation as well? Yes, it does change our amino acids. So we went from phenylalanine to leucine, we went from glycine to tryptophan, we went from alanine to arginine. So yes, it qualifies for missense mutation. Does this qualify for nonsense mutation? It could. It could. What if this codon, this new nucleotide that was inserted, was a stop codon? So if it was a stop codon, the ribosome would stop, and then the amino acid sequence would end here. And then this wouldn't be made, this wouldn't be made, this wouldn't be made. Or in this sense, this wouldn't be made, this wouldn't be made, this wouldn't be made. So that's when a base is inserted in. Well, what happens when a base is deleted, taken out? So here we have the same methionine, lysine. And then here at the same position, well, we were supposed to have UUG that codes for phenylalanine. But what if we take the third base out? What if we take this third uracil out? Well, everything will be moved, will be regrouped after this mutation. So everything downstream from this mutation is regrouped. So now when this U is taken out, the new codon is UUG. Well, that codes for leucine. And then the new codon, the next one is GCG. Well, that will code for alanine. We were supposed to have phenylalanine, glycine. And then here, well, that doesn't code for anything because it's missing a base. So in these two situations, they are also called frame shift mutations because everything downstream after the mutation is regrouped and we are reading the codons differently, that triplet code differently.